decline of fish stocks, loss of biodiversity, and degradation of habitats are just a few examples of the consequences of human activities in the last few decades. And in front of this issue, one of the major tools we have are marine protected areas, which are areas in which human activities that cause habitat alteration or focus on population exploitation are eliminated or greatly reduced. Marine protected areas have numerous potential ecological effects. They can directly affect big piscivores, small invertebrates, or the habitats, and indirectly the whole food web. Until recently, most of the study are focusing on commercially exploited taxa, and only little research has been done on the vagile macrofauna. Two possible models exist. The establishment of an MPA may increase the abundance and biomass of piscivores, which may decrease small carnivores, and in turn, which may increase the vagile macrofauna. On the other hand, the establishment of a marine protected area may increase the abundance and biomass of small carnivores and may decrease the vagile macrofauna. So in this context, the general objective of this study was to assess the potential response of vagile macrofauna and seagrass meadows to different protection levels. So to do that, we selected seagrass meadows, Posidonia sonica, in the Mediterranean Sea, which shelter high biomass and biodiversity of vagile invertebrates. We select marine protected area in Italy, which is composed of 3 protection zones: a totally protected area in red, where everything is prohibited, and 2 partially protected areas in yellow and blue, where restriction decreases. In order to be able to separate the natural spatial variability of this population from the protection effects, we use a special hierarchical sampling design, which consists in each zone of 2 sites, in each site of 2 sectors, and in each sector of 4 replicates. An early sampling device was used for the collection of vagile macrofauna, as you can see in these pictures. Our results showed that the vagile macrofauna was diversified and abundant, and the most important groups were amphipods and mollusks. The application of an ordination methods, that we can see here, allows us to see if the assemblage composition is similar between the different protection zones. So each triangle consists here of a set of samples. And we clearly see a segregation between the set of sample of totally protected area compared to the set of sample of the partially protected areas in yellow and blue, and also the external zone. If we look now at the specific level for the amphipod taxa, for example, we can see that for the totally protected area, the density is low for most of the species compared to the partially protected areas, except for a few species. So we wanted to understand, we tried to understand the factors that could explain these patterns. And one of the obvious factors would be the habitats. We analyzed some statistics on that, and the habitats explain only 0 to 15% of the abundant variation. So we privileged our hypothesis of the predation rate of fishes, which consists in totally protected area. We may increase the abundant biomass of small carnivores, which may decrease the abundance of amphipods. So to do that, we have set up in totally protected area to some exclusion cages to reduce the predation intensity, as you can see in these pictures. And we use this design, 5 total cages to reduce the predation intensity, 5 partial cages to control the cage effects, and some controls which are non-manipulated meadow surfaces. Our results showed that the density of amphipods in this case were higher in the total cages compared to the control and partial cages, which clearly illustrate that the density of amphipods was affected by fish predations. So the take-home message is response to protections are different between macrofaunal populations, and the reasons to see these patterns of the observed patterns are multiple, from the ecological traits of species to the protection-dependent process such as the fish predations. Thank you.